Hello and welcome back to STEM Revise. Today we're going to be discussing the electron microscope, which is video 2 in chapter 2, cells, as part of our A-level biology course. So let's jump right in. So first, let's discuss the transmission electron microscope, or TEM, how it works and its unique features. A TEM uses a focused beam of electrons instead of light to create a highly detailed image of a specimen's surface. The process begins with an electron gun, which fires a beam of electrons onto the surface of the specimen. As this beam scans over the specimen, the electrons interact with different areas in distinct ways. Some areas absorb more electrons and appear darker, while others scatter or reflect the electrons, appearing lighter. This interaction pattern creates a highly detailed image, which is then detected and can be photographed for analysis. TEM images have a unique level of detail that allows scientists to observe structures with remarkable clarity. However, there are a few limitations to keep in mind. Firstly, since the TEM operates in a vacuum, the specimen must be prepared as non-living. Living cells or tissues would not survive this vacuum environment, which means all TEM images are based on dead, fixed samples. This limits our ability to observe living processes directly with a TEM. Secondly, TEM images are produced in black and white. While this doesn't affect structural details, adding color to TEM images must be done digitally afterwards, allowing scientists to highlight certain structures or areas for better visual understanding. Another limitation is the specimen's thickness. The sample must be thin enough or prepared in a way that electrons can interact with or penetrate its surface, which can sometimes be challenging and also time-consuming in terms of preparation. Additionally, SEM images sometimes contain artifacts. Artifacts are distortions or visual features introduced by the preparation process, which can make it hard to distinguish whether a feature is genuinely part of the specimen or just an artifact from the imaging process. Lastly, the images produced by a TEM are usually two-dimensional, giving a flat image of the specimen's surface. Although a TEM provides remarkable detail of surface textures, it lacks the depth of information we might want for viewing internal structures in 3D. So to summarize, TEMs, whilst they are powerful tools for visualizing surface details at high magnification and resolution, they also have some limitations, requiring a vacuum, producing black and white 2D images, and sometimes containing artifacts. They're best suited for applications where we need clear, high-resolution images of surfaces in non-living samples. Okay, so now let's take a closer look at the scanning electron microscope, or SEM, and its features. The SEM is a powerful tool used to produce high-resolution images of the surfaces of cells, tissues, and also small organisms. It's particularly useful for revealing intricate details of the external structure of samples. The way an SEM works is by using an electron beam to scan the surface of the specimen. When the electron beam strikes the sample, it causes secondary electrons to be emitted from the surface. These secondary electrons are then detected and used to form the image. This process allows SEMs to capture incredibly detailed surface features that we might miss with light microscopes. One of the major advantages of the SEM is that the specimen does not need to be thin, unlike with the transmission electron microscope. Since SEMs only examine the surface of the specimen, it can handle thicker samples, giving it a broader range of uses. Another key feature is that SEMs produce three-dimensional images. The way the electrons interact with the surface of the specimen gives the image depth and texture, which helps to reveal the topography and surface details of the sample in a more dynamic way. Although SEM images are initially black and white, they can be digitally colored afterward to highlight certain features or make distinctions clearer. This flexibility in enhancing the image can be crucial for better visual analysis. However, one important limitation is that SEMs also, same as TEMs, require the specimen to be dead. This is because the microscope operates in a vacuum, which living cells or tissues cannot survive. In summary, the SEM is an invaluable tool for examining the surfaces of biological specimens with incredible detail and precision. Its ability to produce 3D images, work with thicker samples, and provide high-resolution imaging makes it essential for many scientific fields. Thank you very much for taking the time to watch this video, where we discussed the differences, advantages, and disadvantages between the transmission and scanning electron microscopes. If you enjoyed, make sure you like, comment, and subscribe. Also, feel free to check out any other videos on this channel. Thank you very much.